Okay, so we're going to start with our processes. We're going to talk about the reactions that these biomolecules are in. There's thousands of reactions that make up metabolism. And we don't want all the reactions to happen all at one time. So what happens is they are catalyzed by enzymes. Is that better? They're catalyzed by enzymes that can be turned on or off. Luckily, we only have a few types of reactions. And so we're going to study those types. But the function of those reactions is metabolism. But metabolism is a huge word. And these are the things that metabolism encompasses. We're going to focus on number one. We're going to focus on getting energy for our biochemical processes. We're going to see how we take one molecule of glucose, monosaccharide, and get our energy molecule, ATP, from that. But we can also study, we're not going to have time, for the synthesis of biomolecules. We also have growth and development, and very importantly, removal of waste and toxic substances. So what we're going to do first is go on this is going to be parallel to our organic little sidetrack. So we studied the organic reactions. Now we're going to apply these to our biochemical reactions. So the first one we're going to study is our nucleophilic substitution. Remember this carbonyl. We see this carbonyl a lot in our biomolecules, and the oxygen is more electronegative. That means that it's going to hog those electron pairs in the bonds, and that's going to leave our carbon slightly positive. Remember that this is lowercase delta. Delta, negative, delta, positive is not a positive charge. It's just indicating that there's more electron density, there's more electrons in this area, and this is slightly positive. So when we say nucleophilic substitution, we have a nucleophile, which usually has lone pairs, seeking out an electrophile, which is electron deficient. So here's an example where a nucleophile, the oxygens on an alcohol on glucose, will attack something that is electron deficient. So this is the first reaction in glycolysis, where we put on a phosphate onto our glucose molecule. This keeps it within the cell. But we're investing some energy here. But you can see that the lone pairs are attacking the partially positive phosphate. See all the oxygens around the phosphate and how the electron density is going to be pulled away from the phosphorus. So, excuse me, we break this bond. So the oxygen lone pairs is a nucleophile, and we're going to call this a group transfer reaction, but it's really nucleophilic substitution. Hydrolysis is another example of nucleophilic substitution. Hydrolysis, very generally, is the breaking apart of a molecule using water. And so the lone pairs on the oxygen and water is going to be doing the attacking. So we're hydrolyzing the phosphate, one of the three phosphate groups, off of ATP. And we're using water as a nucleophile. Again, the phosphorus is the electrophile. Esters, and this is an anhydride, they are amines and anhydrides are susceptible to this kind of attack. All right, we talked about elimination reactions where we're removing a group from carbons that are next to each other. So here we're removing a hydrogen from one carbon and an OH from another carbon, and we're making a double bond. So elimination, we're removing groups to make a double bond. Here, this is a specific type. We call it dehydration. Addition is just the opposite of that, where we are adding across a double bond. And hydration is an example, where we're going to add the hydrogen to one carbon and the OH to another. Those of you that have taken organic know that there's a lot more involved with this, but this is the level that we're going to talk about. Isomerization is pretty straightforward. We're just moving atoms around. There's no net gain or no net loss. We're just moving things around. Now, when an enzyme, in this case phosphoglycerate mutase, is involved, we are usually seeing an intermediate step where something is taken away and then later something is added. So overall, it's just an isomerization reaction, but there's usually, when you use an enzyme to accomplish this, that there is an intermediate step where you're taking something away and then adding something later on. Redox, my favorite, obviously, if I'm asking questions. They are involved in the transfer of electrons. We already talked about that. Remember, Leo goes GERB. We already talked about that. We didn't talk about oxidizing agent and reducing agent. But one thing that's very important is that you recognize that you do not have an oxidation without a reduction. So if you lose electrons, something else has to gain them. And that something else that gains electrons is called the oxidizing agent because it's causing the other thing to be oxidized. So if you're being reduced, you're causing something else to be oxidized. So then you are the oxidizing agent. So if you're being reduced, then you're causing something else to be oxidized, so you are the oxidizing agent. If you're being oxidized, something else is reduced, so you are the reducing agent. Think secret agent. You're on the other side. So again, we're not going to need to worry about oxidation numbers for biochemistry, and we make it really easy. We already went over this. This is oxidation, where we're going to either lose hydrogen or gain oxygen, and reduction is where you gain hydrogen and or lose oxygen. Remember we said that if you're gaining both hydrogen and oxygen, it is not going to be a redox reaction. So it can be either or. Here we see that in this molecule, malate going to oxaloacetate, we're losing 2 hydrogens. We're not necessarily gaining an oxygen. We're just losing 2 hydrogens. So that's why we say and or. Well, if that's being oxidized, something else has to be reduced. And we have what we call a coenzyme that we'll learn a lot about that is doing the gaining. So we have a reduced coenzyme. So this NAD+, the thing that gets reduced, is the oxidizing agent. Reducing, again, is gaining 2 hydrogens and or losing oxygen. Here, what we're doing is we are reducing pyruvate to lactate. You can see that we are going to gain 2 hydrogen. Yes?
It has to be 2 hydrogens.
No, just hydrogen. It can be 1, okay? But it can be 1 or 2. There's different things. So if you end up with more hydrogens, you're in a more reduced state. If you gain up with fewer hydrogens, you're in a more oxidized state. All right, so if we see that pyruvate is gaining the hydrogens, and look, NADH loses 1. So NADH is losing the red one, right, and that becomes oxidized. So here it's just losing 1, and we still call it oxidation. All right, so what's going on here? When you are, so I will, on the exam, give you a reaction and ask you what type of reaction this is. The first one that you should test for is redox. Is it losing hydrogens and or gaining oxygens, or gaining hydrogens and losing oxygens? What's happening here? We're gaining an oxygen. What else are we gaining? Hydrogens. So is that redox? No. What is it? We're going from double bond to a single bond.
Addition.
Addition or hydration. I would take either or, hydration or addition. How about this one? This is sort of not a complete reaction. What's going on here? First, test for redox. What's different between these two molecules? You're losing this one and this one, right? So we're losing hydrogens. So what are we going to call that? Yeah, a redox. It's an oxidation. Don't worry about elimination. I would prefer oxidation. So in your notes, just cross out elimination. I apologize for that. I want you to know this as oxidation. All right, so those are our reactions. Does anybody have any questions about them? Yes?
You were showing the glucose, the ATP for the nucleophilic substitution, like the electron pushing arrow.
Yes. What was the question? In the glucose?
Go back maybe
one more. Right here.
Yeah, on top.
Yes.
Why does that, when the phosphorus gains electrons, why does it push to that oxygen and not from the double bond?
Not from the double bond? Okay, so the question is, why, when these electrons now form a new bond with the phosphorus, why is it going to then break this bond rather than the single bond here? It has to do with the number of bonds that phosphorus likes to make. And it's not going to go from a double bond to a single bond. It's going to go and break this one because of resonance. Actually, resonance is probably the one that's going to dictate this more fully. So it's going to cleave the bond with the rest of the molecule. I am not going to ask you to predict products. Okay? Does that make you feel better?
Yeah.
Okay. I am not going to ask you to predict products. I am only going to ask you to know the reactions and know what type of reactions they are.
Can you go for the oxidizing agent?
This one?
When you just said, like, just the broad term.
Okay, so again, we're wondering why, when the glucose nucleophilically attacks the phosphorus, why are we breaking this bond and not the other bond? When we end up with the product is actually more stable than if we were to have this molecule on this entire thing. That the delta Gs of the products are lower than the delta Gs of the reactants. That is really the main point. That the stability of this is actually quite a strained molecule to have all these negatives together. So it releases energy to break this bond. So this is a lower energy, and then this phosphorus is resonance stabilized within this. This phosphate group is resonance stabilized within this. So it's a more stable group there. But we're going to talk about delta G actually next. But I ask that you don't worry yourself too much about predicting the products. So the last part of this chapter is to get a basic understanding of the genetics or the energetics involved in our reactions. We will look at how the cells get energy in terms of free energy, delta G, and which types of reactions give us this energy. We're going to see how we use this energy. And lastly, before we end the chapter, we're going to see the flow of information in the cell. So let's get started. Very basic. Energy is the capacity to do work. Now, we don't mean go to McDonald's and work a 4-hour shift. Hopefully none of you are doing that right now. I feel bad for you if you are. It is the ability to move matter. Okay, what we're doing is the movement. From all the talk of global warming, we should understand that the light from the sun is our source of energy for all life. If you do not use the energy directly from the sun for energy like plants, then you eat things. that do, or eat things that do, right? So I have Facebook is a funny thing, and one of my Facebook friends is a vegan, and she posts all these things about cows and pigs. So anytime I eat any meat, there's like terrible guilt. Anyway, but many of us consume organisms that consume carbohydrates. We should just consume things with plants, right? We should just eat plants. However, I don't think that's going to change.
For full sustainability, local meat actually works out better.
Yeah, local meat rather than, but it
You end up using land you can't use for plant matter, and you get rid of low quality grain.
Okay. That's good. Then that makes me feel better. Where can you get local meat?
Local farm?
So you have to actually go to the farms? And
You can go to the
Yeah.
farmer's market.
the farmers markets, you can get that? Ours closed down already, so I'll have to wait until the spring.
There's the co-op. There's like the Schenectady farmer's market is inside, and Troy.
Okay.
They open all year.
Yeah. Ours closed down in December, I believe. So, all right. So spontaneous reactions, or reactions that proceed without the input of energy, are those in which the products have lower energy than the reactants. Remember, every molecule contains a certain amount of internal energy. Okay, it's called Gibbs free energy, and it's a free energy of formation, delta G formation. And if the products have a lower delta G of formation than the reactants, then it is going to be a spontaneous reaction. Again, the hydrolysis or the breaking apart of that ATP molecule to take one of those phosphates off, the products have much lower energy than the reactants. The reactions that generate the most energy are our redox reactions. What kind of reactions are in a battery? Redox, yeah. When you took Chem 2, we talked about batteries for a long time. Maybe too long. That's okay. So what value of delta G is going to indicate a spontaneous reaction? Negative delta G. Because remember, delta, that triangle, is final minus initial. So final is a little number, lower number, and initial is a big number. Little number minus big number gives a negative value. So that's why a negative delta G indicates spontaneity. Doesn't mean that non-spontaneous reactions can't occur. They just need some help. And we're going to see a lot of non-spontaneous reactions occur. We're going to do what we call couple them with spontaneous reactions so that the overall delta G is going to be negative. So every time we pass electrons from one molecule to another, we're going to release energy. And we can, again, couple them to those that require energy. All right, so again, we're going to use those redox reactions to make our ATP, our energy currency. I call it our money molecule. Right? It's like money in our pocket, the more ATP we make. Again, more hydrogens in the molecule, it's more reduced, more oxygen, it's more oxidized. So the more reduced molecule it is, the more energy it contains. Think about eating carbohydrates and fats. You would know this if you were alive during the 80s and 90s. But during the 80s and 90s, we were told to eat only carbohydrates, no fats, because the carbohydrates are more oxidized. So we're not going to get as many calories from our carbohydrates. So we were told to stay away from fats. How did that work out? Not so well, right? But fats are from those fatty acids. And remember those molecules had that long hydrocarbon chain, a lot of hydrogens. And look at our monosaccharides, our carbohydrates. They have many fewer hydrogens than our fats do. So I guess fats yield 9 calories per gram, but carbohydrates are only 4. Seems like when you eat a carbohydrate, you never get full. But when you eat some fats, as in the cream in my coffee, then you get some fat and you feel fuller longer. I don't know. That's my thing. That's my rationale for eating lots of fats. All right. So how are we transferring our electrons in biochemistry? We transfer them as a hydrogen with an extra electron. I just want to pause for a moment. And in Chem 2, we studied a lot of acid-base reactions. And acid-base reactions are the transferring of a hydrogen ion of the positive charge, so just a proton. But here in redox reactions in biochemistry, we're also going to be transferring hydrogen, but they're going to have an extra electron. So hydrogens are pretty useful, right? Either they can be just a proton as an H+, or a hydride, H-. So it's very cool how hydrogen can be used in our reactions. So when our molecules get oxidized, then we can see that the coenzymes within our enzymes are going to be doing the accepting of that reducing unit, or our hydride, of the electrons. So they're going to get reduced. And what we're going to do this semester is we are going to take glucose, 6 carbons, monosaccharide, and oxidize it. We're going to burn it. Burning combustion is oxidation. What is the most burned carbon? What is it called? Most oxidized carbon. What molecule is that? Come on, guys. Carbon with as many oxygens as it can. Yeah, carbon dioxide. Carbon dioxide is the most oxidized carbon. When you have a fire in your backyard and you take the carbohydrates and wood and you burn it, you should just get carbon dioxide and water, right? And so carbon dioxide is the ultimately oxidized carbon. We are going to burn, or we are, actually, all of us right now are burning glucose in our bodies. Metabolism is the burning of it, but it's doing it instead of a big fire, and we're not all burned alive, right? We're doing it in stepwise manner. Instead of releasing all of that energy in one big puff of smoke, we're burning in very small steps. And by doing that, we're able to capture the energy. So we could capture it as the heat from burning, stand by our fires, but in our bodies, we do it in very small steps. And that way we can capture, more controlled capture of that energy. And that capture, as that molecule gets oxidized, is going to be where the coenzymes are accepting the electrons. The 2 most common coenzymes that we're going to run into are NAD and FAD. And we're going to look at their structures shortly. Our metabolic reactions can be either classified as anabolic or catabolic. We are only going to be studying catabolic processes, pathways. So they are going to be oxidative in nature. We're going to be breaking down. Anabolic are building up. They are going to be making larger molecules. They're going to be reductive. So I need you to know that anabolic is building up. You're going to be adding hydrogens. Catabolic is breaking down. You're going to be removing hydrogens. And oxidizing, breaking down. So what controls all of this? Well, I said enzymes, which are proteins that can be turned on or off, but how are they made? How are they turned on and off? Well, it all starts with, of course, DNA, which transcribes onto RNA and then translates onto protein. So the word transcribe means just to write over. You're using the same language. nucleic acid to nucleic acid, right? Same language. You're looking at the bases. But when we translate, we're going from nucleic acid to amino acids. Does that make more sense now? You guys going to be able to remember that? Because I am most definitely going to ask a question on the exam between the two of these. Because so many people... Huh? Do you want me to repeat that? So transcription is going from DNA, where RNA is made on a DNA template. So transcription, and the way I remember it, is when you transcribe, or transcription, that is when you're just writing over. So you can transcribe notes by just writing them over. You're using the same language. You're not really changing much. But when you translate from English to Spanish, that's a different language. So we're going from our nucleic acids to our amino acids. Different biomolecule. That's why translate is going from the RNA to the protein. DNA can replicate or transcribe onto the RNA. And then, again, translation is where we synthesize the protein on the 3 types of RNAs. We're going to study this later, but this is a nice picture. Inside the nucleus, we have the RNA polymerase, and I have a really cool video. That's so cool. Where we basically unzip temporarily the DNA. So you have the double helix of the DNA. It unzips, and the messenger RNA, the nucleotides, kind of get sucked in to a little entrance here, and then it's built upon the DNA template. Really cool. So we call that the sense strand and the antisense strand. Translation happens where? Ribosomes. And so the messenger RNA comes along to the ribosome, which is made of protein and rRNA. And then we have our transfer RNA that comes in. Now, which is neat, is you have 3 RNA bases that are called the codons. Am I saying that right? And then on the transfer RNA, which is specific to a certain amino acid, there's an anticodon. And so we build our polypeptide chain by having them come in. So it's sort of like a little, zipping through here, and then the right anticodon on the messenger RNA, transfer RNA comes along. Let's put it all together in our tennis playing lady. Silly. We start with atoms, make our biomolecules, make our organelles, make our cells, make our tissues, make our organs, and then we're off to play tennis. All right, so you're going to be responsible for these biomolecules. I'm not going to start with tissues. Yeah, we're not going up here. We're just going to be looking down in this area. Okay? All right. So let me... Any questions so far? Any more questions? It's review. I'm hoping it's review for most of you. Homework. prokaryotic and eukaryotic cells. I have 4. What is the main one? Yeah. How about size? Which one's bigger? Okay. Which one has organelles? Eukaryotes. And which one can be multicellular? Okay. 2 similarities.
Prokaryotes have circular DNA.
Yeah, they have circular DNA. So yeah, it could be another one. Yeah. They both have the cell wall, and they both have DNA. I mean, that's what I came up with. Ribosomes. Yeah. But where are the ribosomes on eukaryotes? Yeah, on the ER, the rough ER. Yeah. And they're just all throughout the cytoplasm, giving the cytoplasm a grainy appearance. All right. Cytoplasm is goo. It contains proteins. In prokaryotes, proteins, DNA, RNA, ions, small molecules, and it's just stuff in between the organelles and eukaryotes. The cell wall separates the guts from the environment, and beneath the cell wall, you have the plasma membrane that can have receptors. It can detect nutrients and toxins. Our nucleus contains hereditary information. The rough ER, production of proteins. Smooth ER, it is still a area of research, but as far as I know, it's lipid synthesis and biotransformation. Yes.
That's who go from the raw ER to Golgi.
I don't know. I'm just looking for main.
Okay.
Does a vesicle go from the? I don't know. The only one that I need you guys to know the main function of these, because this class had bio chemistry in it. But I'm really, really concerned about what's going on in mitochondria. That's where we're going to spend the entire semester. Yeah, we're going to spend the entire semester in mitochondria. I mean, I need you to know these, and there will be some on the test, but you don't need to know details. You just need to know main function. Which got me through growing cells and isolating proteins from cells and doing all sorts of things. I just knew the main function. So I know I'm biased. And every professor you have will have some bias. And my bias is biology is a tool to get me to the chemistry that's going on in the cell. All right. Biotransformation, what does that mean though? What does biotransformation mean? No?
Is it really, like, I think in the lab, like, bacteria, you're
Well,
transforming them.
what environment are we in? Are we in a non-polar or polar environment? We're in a polar environment. Water is polar, right? So we're in an aqueous environment, and some of the molecules are hydrophobic. So in order to excrete toxins, you have to transform them from non-polar into something that can be excreted. So that's what I mean with the smooth ER. It could mean more things than just that, but that's for the smooth ER, it will take hydrophobic things and make them hydrophilic in order to be excreted from the cell. The Golgi, I hear that it's packaging and distribution. Lysosomes have enzymes, digestion. Same thing as peroxisomes. In plants, they're photorespiration. Mitochondria, energy production, or aerobic respiration. And our cytoskeleton, cell shape, movement, supporting network of fibers. One video I saw that showed it as like zip lines throughout the cell. All right. The endosymbiotic theory is that mitochondria arose when large anaerobic organisms ingested small aerobic bacteria. Aerobic, remember, means that it's using oxygen metabolism. And in mutualistic symbiosis, over time, genes from the small bacteria transferred to the larger cell, and it became the mitochondria. Evidence to support this is the size of the mitochondria, the circular DNA. You guys really need me to draw these structures. But the ionizable hydrogen, make sure that you know where that ionizable hydrogen is. Remember cis and trans. What did I do? I turned that on, right? Do that over. Which one am I drawing now? This. The trans. A lot of people have a tough time with this one. They don't know how to draw the cis and the trans, so make sure that you are good with that. No, I want to erase. No, I want to erase. Erase her. Stop it.
Well, we won't forget it now.
No, you're not going to forget it now. But they've done updates to this. Turn off smartly. It's so outdated. It's so outdated, yeah. All right, now let's try this. Oh, did it go away? It did go away. You guys need me to draw the example? You just look in your notes? You guys are pretty good with this stuff. What do cells use what do cells use the energy for metabolism for? Growth. I had that list, right? I had that list. What are we using the energy for? How did I have that?
I think it was the first slide that we did today.
First slide?
Well, the first slide that we did today.
Oh, yeah. What do we use the energy for? So synthesis of biomolecules, growth and development, and removal of waste. So nucleophilic substitution is when a nucleophile attacks an electrophile to add or remove a group. Elimination is when you're removing groups to form a double bond. Addition is when you're adding a group. In this class, it's going to be where you take away a double bond. Summarization, we're not really changing things, just the position of the group. And redox is the transfer of electrons. And that is the type that is used to generate energy, our redox. And what is the requirement to be spontaneous? Negative delta G. You have messenger RNA that has the information for the amino acid sequence. Transfer DNA RNA carries the amino acid, and ribosomal RNA, it helps make up the ribosome. And then DNA is transcribed onto RNA, and then in translation, the protein is synthesized from the three RNA and our amino acids. Did anybody have any difficulty with the textbook questions? No? You're all good? All right. Make sure you go through them. All right. I'm not going to collect it. No. This is what I'm trying this semester. You know, this is a higher level class, so I'm not going to collect homework. It's not like mastering chemistry where I'm needing to make sure that you're doing it. It's your choice whether you do the homework or not. It's very foolish. This class is not a hard A. If you do the homework, you come to class, ask questions. It's, you know, A or B is, you know, not certain, but highly likely if you do the homework, study, and keep on top of it. So my goal with the quizzes is to keep you guys up on things so it's not a rush the night before the exam. Sort of makes it a little not fun, but. You guys ready for chapter 2? Now, if you guys just took general chemistry, this is pretty much all a review. If it's been a while since general chemistry, then you need to, have laser-like focus. But, perhaps you can add things to help out. Okay, why do we devote an entire chapter to water? It's a universal solvent. Why were they so excited when they found water on Mars? Life. Without water, you're not going to have life. So, as we know it. So, again, this is just a review of Chem 2. And so what we're going to do is talk about the molecule of water. Because when we talk about the intermolecular forces, these intermolecular forces play such an important role in our biochemical reactions and our interactions between the biomolecules. So we really need to pay attention. And we are going to make sure that everyone is on the same page with acids and bases. So there's no student left behind. So we're going to make sure that everyone is on the same page. Again, for those of you that have just had Chem 2, this is going to be a review. So it's a universal solvent. Again, because it's going to transport our chemicals. Just like going to the beach in the summer, it helps maintain a constant body temperature because it has a high what? Heat capacity. High heat capacity. Yes. I'm relying on my chemistry major to answer all these questions. Of course, it's part of our digestive fluids. And remember, I talked about the biotransformation dissolves our excretion products in order to get out of the cell. So what cell part does that? What did I say? Which organelle? The smoothie are. Yeah. Okay. All right. So the hybridization on our oxygen is sp3. You never thought I'd talk about hybridization in biochemistry, did I? Or did you? So why? Because we have 2 lone pairs and 2 bonding pairs. That's 4. That's an sp3 hybridization. Tetrahedral geometry. That makes the molecule bent. Now remember, lone pairs are fat and fluffy, and they take up more space than our bonding pairs. So normally in a tetrahedral, the angle is 109.5, but because these guys are fat, we are going to squeeze the two hydrogen bonds together. And so it's less than 109. So because of this geometry, it's not a linear molecule, but it's a tetrahedral shape for the electron pairs. Water is a polar molecule. What does that mean to be a polar molecule? That's right. One side is slightly positive, not positively charged. It's not ionic. Slightly positive, and one side is partially negative. You have to have polar bonds in order to have a polar molecule. So the polar bonds are the hydrogen and oxygen polar bonds. But if you look at the geometry, water could be nonpolar, right? You could have this, right? But that's not true. Okay, it's not straight. This is not true. It's because of the bent geometry. That is why water is polar. So in a polar bond, you have one atom that is more electronegative and one atom that is less electronegative. Which atom is the most electronegative atom? Fluorine. We don't have fluorine in biochemistry, thankfully. Hopefully not. So our nitrogens, our oxygens, sometimes our sulfurs, we call as our electronegative atoms. So the general trend is that it points to fluorine. The higher the electronegativity of our atom, the more it hogs those bonding pairs electrons. These numbers are made-up numbers. You can't measure these numbers. They're a rating system that Linus Pauling made up. Boy, what a scientist Linus Pauling was. He is the only scientist, I believe, to have won Nobel Prizes in both science and in peace. He got, for his protein determinations, structure of proteins, he got a Nobel Prize. He also got a Peace Prize for lobbying to stop above-ground nuclear testing. I think he passed away not too long ago. And the gentleman that wrote the general chemistry book that we use here had met him. So when he came to visit our campus, I was like, you met Linus Pauling. That is really cool. I just like to be in his presence because he had been in Linus Pauling's presence. So it's sort of like somebody that met, like, famous person that, you know, these are my rock stars. Like, I don't watch the Golden Globes or any of that because they're not rock stars to me. Linus Pauling, like, 2 Nobel Prizes. Then he went a little wacky in his old age.
Don't they all?
You're not saying anything about it. What happened is he started, you know, vitamin C and how people should take, like, large doses. He was nutty about vitamin C. And he went on this kick about, you know, you should have large doses of vitamin C to stay healthy. I think, you know, it's an important part, but it's not the whole thing. And he was, you know, he lost a lot of his respect, you know, people's respect because of his wackiness with vitamin C. But this guy he was in the race for the structure of DNA. He lost out. But he was in that race. He came up with this scorecard for electronegativity, and he won the Nobel Peace Prize. I mean, that is quite the life that I probably won't have. Anyway, so remember, oxygen in biochemistry is going to be the most electronegative. Nitrogen is also going to be electronegative, and these guys not so much. So you can't just have polar bonds. The bond between oxygen and hydrogen is a polar bond. But remember, you have to have geometry. So we have to have a net dipole moment in order to have a polar molecule. Now, dipole moment is something that is measurable. It's measured in Dubai's, and there's a typo on here. What is wrong on this slide? What is wrong on this slide? Yeah, the arrow is supposed to go the other side. So if you guys could correct that. The arrow is supposed to go the other side. So when you are drawing your molecules and you want to show the dipole moment, because it's a vector quantity, vector has both direction and magnitude, the head of the arrow goes towards the negative side. So this one is opposite. And it's probably because the textbook was written by biologists and not chemists. No, what I do is I go through and take things out of the textbook. I write these questions, but I take pictures. I don't draw these pictures. Yeah, I don't draw these pictures. No way. I pilfer from the internet, and from we are given access to textbook images, so I take them from the textbook. Nothing's more annoying than having a lecture that is totally disconnected from your textbook. So I try to take things from the textbook, so you're like, oh, I saw that. I do deviate quite a bit and jump around a little bit, but it's not going to be exact. But yeah, so they made a mistake here. I leave it in here because I like to point out their mistake. It makes me feel good. So, all right, so here, what it is we have really a dipole moment here going from hydrogen to oxygen, another one here, and they add together to go this way. So the head of the arrow is pointing to the oxygen. I don't care about these van der Waals and these bond lengths. I am never going to ask you about bond length. All right, so carbon dioxide also has 2 bonds to oxygens. But why is it not, or why is it polar? Is carbon dioxide polar? No. It's symmetric, right? So you have 2 equal and opposites. This is correct. 2 equal and opposite vectors for dipole moment, and they cancel out. Remember your vector geometry? Yes.
Is that 2 for?
Because there's 2 partially positive because it's pulled from this way. I'm not so concerned about that. I'm just saying that it's pulled this way and pulled this way. And remember your vector geometry. Look, now we're pulling in like algebra stuff. We pull everything together in this class. I love it. So water is truly a unique molecule, mostly because of its ability to hydrogen bond. This property leads to a high boiling point and melting point compared to similar molecules. Hydrogen bonding is an intermolecular force. All right? Yes?
So for water, what would be the difference between boiling and melting?
Boiling, you're going from liquid to gas, right? And melting, you're going from solid to liquid. So that's what the difference is.
Oh, okay.
So hydrogen bonding, and this is what I say in my general chemistry classes, and it's kind of weird. The difference between a covalent bond and an intermolecular force is that a covalent bond, you need chemicals to break down. Your stomach acids, your saliva, the chemicals in your saliva will break down the covalent bonds. But the intermolecular forces you can break with your teeth. You chew ice, when you're chewing ice, you're breaking hydrogen bonds. I'd be really impressed if you could break a covalent bond with your teeth. That would be impressive. But, yeah. But hydrogen bonding is one of the strongest intermolecular forces. And because of that, we draw like little dashes where we see it. And it's the interaction between an electronegative atom, such as oxygen, and a hydrogen that's covalently bound to an electronegative atom. Now, what does that do to this hydrogen? When this hydrogen is covalently bound to this oxygen, look at that hydrogen. That lone pair, the bonding pair right here, where is it? Is it around that hydrogen? No, it's over here near the oxygen. So this is basically like a naked proton sitting out there. And then you have some lone pairs coming along. Those lone pairs are basically interacting with a naked proton. So that's a pretty strong interaction. So remember that our intermolecular forces hold our molecules together. Intermolecular, between molecules. You can break them with little force. So if you crack ice, you're breaking intermolecular forces. And remember, the more that you have, the higher the melting point and boiling point. So think about solid, they're right next to each other. Liquid, they can roll past each other. And gas, they don't even see each other. So going from solid, you have all the intermolecular forces that you can have put together. They're just stationary. They might vibrate in place, but they're stationary. Liquid, you have to break some because they're rolling past. In gas, you have to break all of them. So because of that hydrogen bonding, that's additional intermolecular force, you're going to see that it requires more energy to melt and boil. There are many different types of non-covalent attractions, or our intermolecular forces. And these attractions are either the attraction of positive to negative, partially positive to partially negative, or a combination of these. So the strongest one is our ionic interactions of positive to negative. Salt, right? A salt is any cation, which is positive ion, to any anion, which is a negative ion. Yes?
I've had like 4 teachers tell me covalent is stronger than ionic, and 4 tell me ionic is stronger than covalent.
Covalent is stronger than ionic. And I'm going to show you the data. Okay? The next one is where you take a positive and or negative and interact it with something that is partially positive or partially negative. Hydrogen bonding is then the next. Van der Waals are a collection of our dipole, induced dipole, and our hydrophobic interactions. And we can say this induced dipole is also called dispersion forces. So here, an example of ionic interactions would be table salt, right? Or any other salt, KCl. Ion dipole would be between a salt and water, NaCl and water. Hydrogen bonding, I would say water or any alcohol. Our Van der Waals forces, our dipole would be between maybe our alcohols together or any other dipole. Our dipole, induced dipole, when we have an induced dipole, what you have is something that's nonpolar, but when it comes towards something polar, the electrons get swished around. Our hydrophobic interactions we're going to talk about later, and our induced dipole we'll talk about later. But hydrophobic interactions happen between water and something that's nonpolar. All right, so to put this together and to answer Golan's question about the strength of covalent and ionic bonds, let's take a look at our joules, kilojoules per mole, kilocalories. Let's look at the joules. Covalent bonds are very strong, greater than 210 kilojoules per mole, where ionic interactions are between 4 and 80. All right, so they are definitely weaker. All right, think about an ice crystal, or not an ice crystal, a salt crystal. You can take a chunk of salt and break it apart, and that you're breaking ionic interactions. Hydrogen bonds are then next, and our Van der Waals are much weaker. Hydrophobic interactions, again, I'm going to talk about later. So how much time do I have left? 8 more minutes. I'm going to ask that you guys stay after I stop recording so that I can take attendance. Okay. So let's start. with our ionic interactions. And what we're going to do is we're going to apply them, because I know that you guys have learned about these before. I'm going to apply them to our biomolecules. So when do we see ionic interactions? Again, they occur between actually charged atoms or groups. So you have to have an actual charge. It has to be positively charged or negatively charged. In proteins, what we have is ionic salt bridges. We have amino acid side chains that are either acidic or basic. And if it's acidic at neutral pH, it's going to be negatively charged. If it's basic at neutral pH, it's going to be positively charged. So here I'm showing the side chain of an acid amino acid, the R group, and here's the R group of a basic one. And if this protein is at neutral pH, then the acid will have donated its proton and the base will have accepted a proton. So the acid's going to be negatively charged and the base is going to be positively charged. So we call it a salt bridge. Salt bridges are very important in our protein interactions. What would happen if water were present around this? It would have donated the hydrogen to water, hydrogen ion to water, but wouldn't water surround the oxygens of water, the hydrogens of water surround the negative part? And wouldn't the negative part of water surround this positive? So where do you think we see the salt bridges in our structure of our protein? You see it in the interior or the exterior, these salt bridges? Interior. Because when these negatively charges are around water, or the positive charges are around water, they get hydrated. So here, pretend this negative charge, this chlorine, is that negative amino acid. Then the positive side of water is going to surround it with a shell of hydration. If you have the positive, say the positively charged basic amino acid, then the negative side of water is going to surround it. This is what happens when you dissolve salt in water. Okay? The salt crystal dissolves. And why it dissolves is that it's more stable to have the sodium surrounded by the negative side of water and the chlorine surrounded by the positive side of water. Do you have a question? Go on.
Yeah, so these salt bridges are going to form inside the water.
Yes. The salt bridges are going to form in the interior of our protein. So say this is a negative acidic side chain, and this is a positive basic one. What they're going to do, and my whole body is the amino acid chain, they're going to fold up so that they're away from water and they can interact together inside. I love protein structure. Again, hydrogen bonding is weaker. We see hydrogen bonding when we have hydrogen covalently bound to what?
Fluorine.
Oxygen, fluorine. We don't see fluorine. Oxygen or nitrogen, basically. And then it's going to interact with a lone pair. And so what we see with water is that it can have 4 hydrogen bonds. That's a lot for one molecule. That's a lot of hydrogen bonds for one molecule. And what happens is remember, when you're a solid, you have the most intermolecular forces you can possibly have. So when you get 4 hydrogen bonds per water molecule, you get this 6-membered ring that looks like this. And it forms our ice crystal. What's in the middle? Nothing. Empty space. Nothing. And so what happens is when you go from liquid water to solid water, you actually create more empty space. And that is why the solid water floats on top of liquid water. You would think that the density of ice should be greater than that of liquid water, right? But it floats. So that's because of this 6-membered ring. And so because of all that hydrogen bonding, you have a really interesting thing that happens with the density of water, and it affects, of course, the melting point.